All right, awesome. We're going to go ahead and get started. So welcome everyone, and thank you for joining PARC's Disease Task Team webinar, Protistan Pathogen Perkinsa webinar. My name is Jenna Palmisano. I am the chair of the National and Southeastern Regional Disease Task Teams, and I'm a PhD student in Dr. Anna Savage's lab at the University of Central Florida. I primarily study pygmy rattlesnakes and the impacts of emerging pathogens on them. So we are pleased to kick off today's webinar. Our goal with the webinar series is to help connect science and management action by providing information to land and natural resource managers. And so we welcome input and feedback and any questions that you have. So before I introduce our speakers today, just a little bit of housekeeping. Captioning is available by clicking the CC button at the bottom of your screen. This webinar will be recorded for later posting on our PARC Disease Task Team webpage and YouTube, and all attendees will be muted until the Q&A session at the end. And if you have any questions, you can ask them via chat during the presentation, and myself and Kobe and Kerry will be recording those so that we can ask them at the end. All right, so to introduce our speakers, Dr. Vanessa Smilansky is a research scientist in the greater Boston area. Her interests include pathogen genomics and the use of omics-based biotechnology to control the spread of wildlife disease. And Dr. Matt Atkinson, our second speaker, is a lecturer at the University of Central Florida and the new junior co-chair for the Southeastern PARC. His research interest focuses on amphibian ecology and conservation, with a focus on pathogens and incorporating immunogenetics, population management, invasion biology, and urban ecology to answer questions regarding the future of amphibian populations in the Anthropocene. So on behalf of PARC, we would just like to thank PARC's Disease Task Team for helping to develop a variety of products mentioned in this talk, as well as disease-specific fact sheets available at the Disease Task Team website. And of course, we especially thank our speakers today. So I'm now pleased to introduce our speakers, Dr. Vanessa Smilansky and Dr. Matt Atkinson. So Vanessa and Matt, please take it away.
All right. Well, thank you. Thanks for having me. Appreciate it. I'm going to begin with a global and evolutionary overview of the Perkinsa group of parasitic protists. Taxonomically, they're best defined as a class within the phylum Perkinsozoa within the superphylum Alveolata. There's a few other alveolate groups that are known to infect frogs, primarily found in intestinal tissues. These include putatively commensal extracellular ciliates and intercellular apicomplexan subclasses Coccidia and Gregarines. Coccidia have been linked to disintegration of intestinal epithelium, but none of these groups have been linked to mortality or interference with metamorphosis. Perkinsa, however, have been extensively linked to disease and mortality events in frogs to the point where severe Perkinsea infection is called the third most common infectious amphibian disease. Oops, sorry. After chytridiomycosis and Rana virus. So Perkinsa are a hugely diverse group. They parasitize many types of hosts, not just amphibians. Last year, Metz et al. Assembled an updated Perkinsa phylogeny from amplicon sequencing variants, which were generated from a meta-analysis of a global eDNA dataset targeting the 18S rRNA gene. The phylogeny revealed 4 environmental clusters, as well as the major known clusters of parasitic Perkinsa. So these include Parvulaciferaceae, which are parasites of marine algae. They're an interesting case because both the host and the parasite are single-celled and similarly sized. So the parasites actually take up almost the entire host body before emerging and eventually killing the host. Zelidae are parasites of fish, which can cause large xenomas in the gills. Perkinsidae are parasites of mollusks, which can cause dermo disease and economically important oysters. These parasites were among the earliest Perkinsa studies, which date back to the 1940s. And then finally, there's the novel alveolate group 1, or NAG1, which is the most evolutionary derived Perkinsa group. And this is where we find the infectious agents of frogs, so which are linked to severe Perkinsa infection, primarily in tadpoles. Perkinsa have very complicated life cycles. They typically include a free living stage and an endoparasitic stage. These life cycles have been observed in culture. However, amphibian Perkinsa has proven very difficult to culture so far, but other species like Parvilicifera cinere and Perkinsa olseni have been successfully cultured. And so we can speculate that the amphibian Perkinsia life cycle is similar to P. olseni based on their close phylogenetic relationship and the fact that certain life stages like the trophozoite and the hypnospore have been observed in amphibian tissues. So in a nutshell, the hypnospore is released from infected hosts into the environment, where it develops into a zoosporangium and releases myriad flagellated zoospores, which then go on and infect new hosts and develop into these trophozoites. So the trophozoite is the feeding stage, and it's the one that's typically associated with disease symptoms. So Perkinsia are found, they're really everywhere. They're found in a variety of habitats. Historically, they were associated with marine environments, primarily because of dermo disease and the economic impact that it had. Amphibian mortalities linked them to freshwater, and then last year's Metz et al. Study revealed a surprising diversity in soil. So each of these plots here represents a main habitat, and they show the connections between different habitat zones on the top half, and the connections with the taxonomic groups on the bottom half below the dotted lines. So the width of the connectors corresponds to the number of amplicon sequencing variants in each taxon. And the NAG1 group, which of course includes amphibian Perkinsia, is this gray-blue color. So in marine habitat, NAG1 was primarily found in epipelagic and estuarine zones, and in soil habitat, it was found primarily in temperate and tropical forest soil, and in land water habitat, it was found primarily in lakes, rivers, and freshwater sediment. So this study shows ultimately that NAG1 is more widespread than previously thought, because up until this point, it had only been found in freshwater habitat. So really interesting study. This is a previous study from Chambouvet et al. That shows a detailed distribution of NAG1 sequences, also based on the 18S rRNA gene. This group includes environmental sequences and sequences derived from both healthy and diseased amphibian tissues. Sequences from healthy tissues grouped into distinctive clades that were almost exclusively found outside of the US, in Europe, Africa, and South America. It's unclear if these clades should be considered non-pathogenic because most of the tadpoles were sampled from early Gosner stages, possibly too early to detect any disease progression. And at least one sequence from clade B was linked to the 2006 mortality event in southern leopard frogs in Georgia. Most of the sequences from this event grouped into a separate clade, which was named pathogenic Perkinsia clade, or PPC. Since 2006, PPC has been linked to many outbreaks of disease called severe Perkinsea Infection, or SPI, and more recently, it's been linked to cryptic infections with little to no signs of disease. So most of the PPC-related disease outbreaks and mortality events have occurred in tadpoles in the US, which date back to 1999. In 2021, a mortality event in captive-bred European tree frog tadpoles was recorded in the UK, and then in the last few years, PPC has been detected in cryptic or sublethal infections of tadpoles in the southern US and Panama. And then interestingly, it was detected in these high-altitude terrestrial breeding frogs in Ecuador. So these frogs don't actually have an aquatic tadpole stage, which means that PPC is likely being picked up in tropical forest soil, which, as we saw from that Metz study last year, it's known now to be a habitat for Perkinsia. So I thought that was a really cool connection. In 2021, my group at the University of Exeter, where I did my PhD, created this NAG1 phylogeny that expanded the diversity of PPC. We included sequences that were collected from cryptic tadpole infections in Panama and those collected from the mortality event in the UK and the tree frog tadpoles. And we compared them to previously published sequences in the US, and we found that they were almost genetically identical. And we also found that they were genetically distinct from those putative non-pathogenic clades, A, B, and C, that had primarily been found outside of the US. So this was the first published report of PPC in cryptic infections of tadpoles, which indicates that PPC pathogenicity is not absolute, and it likely depends on dynamics between the host, the parasite, and the environment. Also, for our 2021 study, we were unable to identify the Panama tadpoles morphologically, so we sequenced a portion of the mitochondrial 16S rRNA gene and assembled the sequences into a phylogeny. Of the 81 tadpoles that we sampled, 10 of them were positive for PPC, which are highlighted here in red. They were distributed across 3 families, a Hylidae, a Romobatidae, and Bufonidae. This was the first recorded observation in Bufonidae, which is a hugely diverse group of toads. And we detected PPC in Rana marina, which is the cane toad. So Central America is actually the native range of the cane toad, but outside of this region, it's been a common invasive and potential pathogen spreader. So it was kind of alarming to find this cryptic PPC infection in this species. So to conclude the global perspective of Perkinsea, observations of infections in amphibians suggest that Perkinsea virulence is fluid and dependent on the interaction of the host, parasite, and environmental variables. This diagram illustrates the interaction of these variables, and I've broken it down based on the global Perkinsea observations. PPC is a specific parasite genotype that's associated with virulence. It's genetically distinct from other Perkinsea clades, not typically linked to virulence. The trophozoite stage of the Perkinsea life cycle is the parasite phenotype linked to virulence. Host genotype seems to influence virulence because certain species are more prone to Perkinsea morbidity and mortality, such as the dusky gopher frogs. This is something that Matt's going to get into in more detail. The tadpole life stage is a specific host phenotype that is more prone to Perkinsea infection, morbidity, and mortality. And this is likely due to tadpoles having a less developed immune system. And in some species, it's actually been observed that infected tadpoles metamorphose more quickly, possibly to help clear infection. And then finally, environmental variables strongly influence virulence, and these might be the most difficult to understand. So far, it seems that Perkinsea outbreaks and mortality events are associated with cooler months, possibly due to an increase in host availability during breeding season. And Rana virus co-infection could be a synergistic strain on the tadpoles' already underdeveloped immune system. So Matt has some more to say about this, and so I will let him take it away.
All right, perfect. So let me go ahead and swap here real quick. Just to do a quick double check, is everybody able to see my slides? Perfect. All right. So while there does seem to be some instances where we do see these Perkinsea infections outside of the US, for the most part, a lot of the current research is focused primarily in North America, mostly because that's where we're seeing documented mortality events a lot more consistently. Now, it's kind of an interesting history of Perkinsea, and one thing I like to point out is that we have a fairly good idea of when we first documented these kind of initial mortality events. They actually occurred in New Hampshire in about 1999, which for context is very, very similar to the timeline that we discovered that BD was a very strong influence in the kind of amphibian declines that we've been seeing globally. Around that time, we discovered that there were a number of events that were widespread across the US, documented through a couple of different studies, including Davis 2007 that Vanessa mentioned a little bit ago, and that it could affect species of special concern, particularly our lovely friend down here, the dusky gopher frog. However, there's been a lot of questions about taxonomic identity, and given at the time the pathogen was kind of deemed to be this highly specialized opportunist, they kind of considered that this was likely not an important threat of amphibians. And to be honest with you, we kind of moved away from really focusing on this pathogen outside of a couple small groups for most of the 21st century. However, more recently, there's been a number of really awesome screening abilities that have been created as a result of just kind of renewed interest in this pathogen, particularly due to some of the interesting interactions that were happening with the dusky gopher frogs. And as a result, it allowed us to greatly increase our ability to detect Perkinsea infections in wild populations, particularly targeting those more widespread cryptic infections that may be more common, but you don't necessarily always manifest into a full mortality event. Specifically, SPI infections were found to be fairly widespread across a massive variety of different hosts, especially compared to what was originally anticipated. And looking at swab museum specimens, this documented SPIs occurring as far back as the 1920s in Florida, and likely probably farther back than that. That was just the last time period that there were samples for that were swabbed. But unfortunately, a lot of SPI research is still pretty heavily in its infancy. We don't know a ton yet. And while we do know some things about the just basic disease ecology, there's still so many more questions that we have. And ultimately, that's kind of a running theme with a lot of the things that I'm going to be talking about today. So what exactly do we know about Perkinsea? Because while we don't know a ton, we can still kind of start making some inferences that can ultimately help us improve our management actions and ultimately both in situ and ex situ, and hopefully try to augment populations going forward. In general, we have a decent idea of a couple very specific points. First is that through work through Josh Cook, we have a fairly good idea of how the transmission of this pathogen occurs between kind of sessile hypnospore stages to active zoospores colonizing hosts and ultimately potentially killing hosts. And it appears that there may be even more than one ecotype, which makes sense given the kind of diversity that we see from a genetic perspective at that NAGO1 clade. It also infects a wide variety of different hosts across broad taxonomic and ontogenetic breadth, so both tadpoles, metamorpho, and adults. And we see that fairly consistently across a wide variety of different studies. Perkinsea infections are highly influenced by environment, so a lot of factors, both abiotic and biotic, do seem to play a role here. And finally, that co-infections with both chytrid fungus and ranavirus are relatively common, which is kind of concerning. So to kind of dig into these in a little bit more detail, let's start first with transmission. Perkinsea is highly opportunistic, infecting hosts through a variety of different pathways. For instance, they can have direct colonization of the host by a free-swimming zoospore stage, where that will actually burrow into the skin and ultimately colonize the intestinal tract and kind of the kidneys, the liver, and a couple of other different places. However, that tadpole may also pick up zoospores through ingestion, and this again can also include the hypnospores as well. This can occur through incidental filtering of the substrate by those tadpoles as they're just kind of looking for algae and other small things that they're trying to consume. It could be also potentially facilitated through the consumption of either invertebrate vectors or scavenging on other amphibian hosts. So we do see that there is some instances where cannibalism does occur in some of these tadpole species. So we expect that if an infected animal is cannibalized, we expect that animal could be potentially infected through that pathway. Now, unfortunately, Perkinsea hypnospores can be incredibly difficult to kill, which makes their transmission rather easy. Through some anecdotal data, they can be able to survive through prescribed burns, survive long periods of desiccation with getting well over a year of having no water in contact at all, can survive through the intestinal tract of birds, so in other words, dealing with all the stomach acids and be able to completely pass through as a hypnospore stage and still be infective. And this may actually facilitate the movement of this pathogen from one location to the next, which makes it really difficult to control in some instances. To this point, we only know of about a 10% bleach solution being effective and long-term freezing as kind of opportunities to completely destroy and inactivate those hypnospores. And again, that's all through kind of anecdotal data and stuff that came out of Josh Cook's thesis from 2008. Ultimately, this means it's basically nearly impossible to completely remove Perkinsea from the landscape. So it's going to be, from a kind of management perspective, more important to focus on the kind of overall, how do we control from an augmentation versus decrease, because you're never going to completely get rid of it, as well as where there are areas that are currently not having those existing SPI infections. It means that we need to be incredibly careful about biosecurity going into those locations. Interestingly, we do see that there's some sort of ecotype differences between these Perkinsea strains, and while that may be genetically related, it could just be local ecological adaptation. Ultimately, this is based off of anecdotal data that showed that there were kind of these different ecotypes that exist based off of type of wetland. Where you had more permanent wetlands, this Perkinsea spore would be colonized primarily things like bullfrogs and pig frogs, and it would be slightly less pathogenic. Whereas the ones that were showing up in more ephemeral wetlands that were targeting more Sphenocephala, so southern leopard frogs, and the dusky gopher frogs, Florida gopher frogs, that were more likely to exhibit high infection intensities and mortality, but both could still do it. So that's an important point to note. So again, it's these ecotypes differ primarily based off of their mortality and intensity rates, but again, both of them were still able to exhibit these particular interactions. And this kind of complicates our ability to really do a lot more research on this pathogen. We need to start kind of disentangling the taxonomy and how these ecotypes fit into that, so we can actually start kind of drawing the relationship between functional and genetic information. So digging into actually how this pathogen affects those amphibian hosts, we're going to first start with life stage. We find that there's a ton of variability in mortality, infection intensity by life stage. Most of the actual high intensities in mortality are typically observed in tadpoles. And again, this is both through a number of different studies, or that it's happening close to metamorphosis. So basically, right as they're getting out of that kind of tadpole stage, that's when we're seeing that bit of mortality. However, understand that adults can still exhibit high intensities and mortality. So just because you metamorphose doesn't mean you're quite out of the woods yet. However, there's no typical difference in prevalence between the larvae and the adults. So in other words, the roughly same amount of them are infected, but there's differences in the strength of infection or how likely you are to progress into disease, and not the actual number, the likelihood of infection itself. This likely indicates that all can become infected and experience mortality, but larvae are a lot more vulnerable to this pathogen, which is a lot more concerning. And so there is some data that does back this up. This graph from Smith et al. 2021, you can see that upon the initial deposition of the egg masses themselves, survival is fairly high, but as you start getting into those very, very early stages of larvae, it drops off rather significantly. And then ultimately it starts building back up kind of as those larvae get a little bit older. Now, as far as taxonomic diversity goes, outside of the one family that I did not include on here by accident from Central America, we've seen pretty much almost all of the host families that have been tested for in North America showing some sort of infection. Buthaudidae, so we've also seen it in southern toads and Gulf Coast toads, so two different genuses there. Ranidae, so our leopard frogs, bullfrogs, pig frogs, gopher frogs, pretty much anything that's a rana we've seen it in. And it's consistently kind of our more problematic group when it comes to these things. Hylidae, so your tree frogs. Pipidae, which is a rather strange group of kind of primitive frogs that we have, unfortunately, as an invasive species here in Florida, and as well as in a couple other places in the US. Eleutherodactylidae, a direct developing frog, so again, a species that's not going to actually have a tadpole stage, which kind of makes that post-life stage stuff a little bit more interesting. Microhylidae, so our narrowmouth toads, and surprisingly enough, Salamandridae. So our kind of newts and that sort of grouping. So it's not just in frogs, which is even more interesting. And again, there's many more that are also likely, but just haven't been tested yet due to just lack of funding or lack of just interest at this point. However, while all these different families can exhibit infections, we do see massive differences in prevalence. So our ability to, whether or not those animals are going to be infected, intensity, how strong those infections are, and ultimately mortality. Now, many host families are known to only currently exhibit that cryptic infection, so things like our Salamandridae, Microhylidae, Pipidae, but that may just also be kind of a product of how many people are looking for this at this point. However, consistently we find that Ranidae typically exhibit really high prevalence, really high intensities, and consistently are the ones that are going to show the most mortality events. And again, this is across a wide variety of different studies that we find this. And unfortunately, this especially affects the gopher, crawfish, and leopard frog clades, which are numerous imperiled species that are a part of that group, which is concerning. Now, what's even more interesting, though, is that many of these invasive species, our lovely Cuban tree frogs, American bullfrogs, and pipids, so our Xenopus tropicalis here, are also documented hosts, which can come with a lot more dynamics that we have to be kind of mindful of as well. As far as how the impact of the environment kind of alters Perkinsa infection dynamics, we see that prevalence and infection intensity is very location dependent and is influenced by likely a variety of different factors, both biotic and abiotic. Perkinsa infections tend to exhibit very strong seasonality and appear closely tied to kind of variances in precipitation and temperature, as well as there may be some interactions between things like urbanization or just habitat modification more broadly, which may affect these infection dynamics. However, we still need a lot more data on this. Most of our kind of thoughts on this come from some preliminary comparisons between kind of classic Florida ephemeral wetlands versus these wonderful things that are rather common here, these little retention ponds that are kind of what's left of most of these ephemeral wetlands in Florida, unfortunately. And what's even more interesting about that is that these kind of urbanized sites exhibit fairly low SPI infection rates, but is that a product of just kind of the biotic community that's using those locations or just kind of the environmental parameters? We're really not sure yet. As far as kind of the abiotic components of this, we do see that there are differences in prevalences across species and families as far as overall infection rates. And that does seem to alter the overall community SPI infection dynamics. So in other words, if you find a lot of randed frogs and they're the ones that are most likely to be infected, we do see a strong increase in the likelihood of infection by other species in that location. So in other words, even if you're not a randed frog, if you're sharing a habitat with a bunch of them, you are more likely to then exhibit Perkinsea infections. They may be cryptic, they may be low-level infections, but then you kind of serve as a reservoir for other things in that environment. And there are a couple other species that seem to be impactful with this as well. Things like Gastrophynes, or our narrow-mouthed toads. Out of all things, that was one of the highest things as far as kind of influencing the Perkinsea infection rates across the community, which is rather bizarre. It was not something we were expecting when we did that paper. And even more concerning is we have our lovely Xenopus, an invasive species here in Tampa, that when they do show up in wetlands, it appears that they are likely to increase the infection prevalence in the native species outside of that context. So here in that bottom panel, you see a figure from my dissertation where you have residents in green from invaded sites, residents from never invaded sites, and then the Xenopus tropicalis themselves. And you'll see that the residents from invaded sites have significantly higher prevalence than both the residents in never invaded and the tropicalis themselves. So even though the tropicalis might not be affecting it a ton, there does seem to be some sort of interaction there that we should be concerned about, where maybe these tropicalis are serving as either amplification or reservoir hosts. Now, to get into the seasonality component a little bit here, based on our somewhat limited data, and granted, most of this is based off of the southeastern United States, there are some fairly strong seasonality patterns, where here in Florida, we see fairly high prevalence in the kind of January through March timeframe. It drops off to the lowest point around June and July, and then starts picking back up over the summer, ultimately kind of going back up to what it was around December to get back to that January, February, March timeframe. Most specifically, we see that these infections occur primarily during those cooler months for us around February and March. But bear in mind, cooler for Florida does not necessarily mean cooler for everywhere else. For us, that's around an average temperature of about 70 degrees Fahrenheit. However, spikes in prevalence and intensity do occur outside of these temperature patterns and appear to be tied to higher than average precipitation. You may notice that in particular in this plot, you see a rather large spike in September. If you were to break that out by year, most of that is attributed to a single month in the year of 2017, which is when we had Hurricane Irma come through and actually decimate parts of Florida. And we saw Perkinsea infections right after that, which is kind of an interesting phenomenon there. Unfortunately, that's highly anecdotal, so we can't say a ton about that yet. So to kind of dig into that a little bit more, we do see that infections appear fairly closely tied to increased precipitation, with increased mortality observed kind of post-rain. So here from Atkinson 2016, which is my master's thesis, we find that when we get large rain events, it would change the water level and also spike the pH. And around that same time, we lose a fairly high percentage of the remaining population of those tadpoles at the time. And that is, again, a significant relationship. And additionally, this was further reinforced by the finding that we found increased prevalence and intensity during months with high precipitation in Atkinson and Savage 2023. This may be a very interesting evolved relationship where spores have evolved the ability to increase their hatching rate as they get closer to this pH of around 6.5, which for context, for a lot of these ephemeral wetlands are usually around 4 to 5 in general, as from a pH perspective. And so when you get these large, massive increases in rain, it will bring the pH of that wetland up. And as a result, it kind of triggers that hatching rate. This was observed in dusky gopher frogs again after those particularly in more extreme and flashier wetlands that are going to fill really quick and have a lot more of that rapid influx and change in pH. However, it's more tied to that temporary increase and not to the long-term pH. In other words, if that animal was living permanently at a pH of 6.5, a pH of 7.5, it did not change its likelihood of mortality, only that rapid shift of pH. And that could be related to stress issues as well as the actual just kind of prompting of those spores to kind of colonize new hosts. Now, finally, and kind of even more importantly, I think we need to take a stock of how does this particular pathogen interact with other pathogens. We see that there's some really, really strong connections between Perkinsea and ranavirus across a wide variety of different studies, including a couple that have documented mortality events from both Perkinsea and ranavirus simultaneously, where some individuals are slightly more infected with Perkinsea, some are slightly more infected with ranavirus, and it kind of triggers a big mass mortality event across the population. Particularly, that was observed in Landsberg 2013 here at a site that we have here in Florida. And based off of studies from Atkinson established 2023, infections by one pathogen appear to increase the likelihood of the other. So in other words, if you're infected with Perkinsia, you're more likely to be infected with ranavirus. And if you're infected with ranavirus, you're more likely to be infected with Perkinsia. However, there may be differences on the impact of the host though. Cuban tree frogs infected with Perkinsia and co-infected with both Perkinsia and ranavirus exhibited loss in body condition, whereas individuals infected with just ranavirus did not. So there may be some interactions there. As far as BD goes, we do see that Perkinsia and chytrid can occur as co-infections as well. And there's some indications that one pathogen is benefiting the other, but it's difficult to tease apart due to these really close ties and seasonality for both pathogens, where both tend to prefer these cooler temperatures here in Florida. And so we need a lot more data, particularly experimental data, to say anything, unfortunately, this time. And what's even more concerning is that we do see that there's even instances of tri-infection across BD, ranavirus, and Perkinsia, and it's been documented a number of different times. So ultimately, the point that I'm trying to make here is that we really need to start thinking about amphibian pathogen dynamics using a community dynamics perspective or a One Health perspective, instead of just isolating and thinking about just Perkinsia, just BD, just ranavirus. So why should you care about Perkinsia? So currently, Perkinsia infects a wide variety of different protected species, everything from federally listed species such as the dusky gopher frog that we'll get into a lot more detail on here in a little bit, which is lovely guy up here, as well as the striped newt. Though this might be cryptic infections only, we need more data, but this was just recently reported by Eric Hartman's group out of the University of Florida. So that's a really concerning development that we're not super happy about. But outside of federally listed species, there's a number of state-listed species as well, both the Carolina and Florida gopher frog, which is state-listed throughout much of its range, as well as the crawfish frog, which is, again, state-listed throughout much of its range. So to kind of zoom in here really quick on dusky gopher frogs, these SPI mortality events have likely contributed to the decline in this species, with numerous years of little to no recruitment despite conditions appearing beneficial for recovery. And this was documented across a couple different master species, including one by Jamie Smith from 2021, and my own in 2016. And unfortunately, there's strong evidence of very strong different impacts on the head starting efforts themselves. However, there is some good news. It does seem like older tadpoles may be less susceptible, and as a result, we may be able to kind of curtail how we're doing head starting to where it can actually still be beneficial in this particular instance. We also see that there's no evidence of acquired immune response in this pathogen. This may be related to the fact that dusky gopher frogs, at kind of their last that I checked, there are roughly around 200 to 400 adults left in the wild. So as you can imagine, there's a fairly strong effect of population bottlenecking at that point, which may kind of be influential in this regard. There is a lot more research that's being done on the host immunogenetics for the species in comparison to Capito, as well as comparison to Sphenocephala, which is kind of, or so southern leopard frog, which is kind of its most closely related outside of the gopher frog clade. And as a result, there's now active work to better monitor these translocation sites for Perkinsia, so that way we can start kind of making progress on finding where these species or where this pathogen is showing up in really high numbers, trying to avoid those locations, as well as what's happening to our translocated populations for this species. But it's not just the imperiled species that we have to be concerned about here. SPI impacts also a lot of invasive species, which can result on subsequent impacts on native species. So we'll focus in on the American bullfrog and Cuban tree frog, both of which are fairly common invasive species, depending on what context, different parts of the US. As well as we do see that greenhouse frogs and these western clawed frogs both show infections and potentially fairly high infection intensities. But at this time, they're fairly locally isolated, and we just don't know that much yet. So to kind of zoom in here on bullfrogs, the species has already been well documented to serve as a reservoir and amplification host for ranavirus and BD. That was in a review paper that mine and my advisor came out with this past year. And given that they are regularly infected with Perkinsia in both their native and invasive range, including exhibiting mortality events across both their native and invasive range, this may indicate that bullfrogs could also serve as a really good effective reservoir or amplification host for Perkinsia. However, at this time, we do not have current experimental data on this. So there's some really awesome opportunities here for somebody to dig into these interactions a bit more. Now, Cuban tree frogs, I think they're a really awesome, interesting interaction. And we do see that they are regularly infected with Perkinsia here in Florida and in Louisiana, which is their kind of other major hub at this point. But I do want to point out that the species is rapidly moving across the southeast, and we're kind of getting reports of populations in Georgia, South Carolina, as well as in Mississippi. So we do need to be kind of mindful of that. And while Perkinsea infections do not for the most part not seem to affect them that much, we do see that there are some negative impacts on these hosts with decreased body condition, like I mentioned earlier from that New Orleans study, as well as increased metamorphosis rates, where these animals are clearly trying to metamorphose quicker and try to get out of these wetlands as fast as they possibly can, although there's still a lot more that we can dig into this. So ultimately, these impacts of SPIs on conservation and management are likely to get worse. Ultimately, other stressors, whether they be genetic stressors like population bottlenecks, environmental stressors, global climate change, all could potentially exacerbate SPI infections and kind of shift those current interactions where SPI is only really causing these cryptic or mild infections into that disease and ultimately mortality state. So we have to be incredibly mindful of where we're kind of dealing with this problem. Specifically, we see that global climate change may increase the likelihood of favorable conditions for SPIs outside of Florida and in Florida as well. And that tolerant invasive hosts might also likely be continue spreading this pathogen around. So we may have to kind of figure out how to deal with that. However, there's some things that can be done to lessen these impacts of SPI on the landscape. The first of which is just better biosecurity practices. Even though we're never going to fully remove this pathogen from the landscape, it's just good practice to make sure we're not augmenting it at the same time. Use gloves when handling any amphibian and disinfect field gear once you return from the field. This is especially for areas where Perkinsea is not currently documented, because you really don't want to be moving this into a new area when you really don't have a good likelihood of being able to remove it from that point on. Currently, we do think that about a 10% bleach solution, and bear in mind this is 10% pool bleach, with about a contact time of 5 minutes, is the only really solidly good way to destroy those hypnospores. The PARC DTT has developed numerous guides on how best to do this. I would really highly recommend y'all taking a look at those. There's some great information there. But to get more into the management aspect of it, I highly promote that you should probably be removing invasive species from these more vulnerable areas. Many of these invasive species, as I've already been documented here, between Cuban tree frogs, American bullfrogs, and our western clawed frogs, appear to be harboring rather high infection intensities and high prevalences of this pathogen. And by removing these individuals, we remove a major source of infected Perkinsea spores from the landscape. So as much as we don't really want to remove these invasive species, and it is kind of easy to kind of fall into the trap of saying, well, they're just kind of part of the ecosystem now. Unfortunately, this kind of puts more of an emphasis on we probably need to be doing a little bit more. And finally, there's also a decrease in other pressures on those native hosts. So if you remove those invasive species, you're removing that kind of competition element, at least a little bit more, and allowing those native hosts to not be as stressed, and so they're less likely to kind of progress into those disease symptoms. If you're doing head starting, you should probably cater it to avoid Perkinsea and really any other pathogen. Have a good understanding of the site that you'll be releasing individuals to, avoid flashy locations with a lot of ranids, and screen for pathogens prior to release, particularly Perkinsea, but as well as a number of others. You should release older, more established individuals to ensure higher likelihood of survival, particularly given the information that we got out of the Atkinson 2016, my thesis from them. And again, follow these better biosecurity approaches mentioned earlier. And finally, it is really critical to continuously monitor these sites for potential outbreaks and reassess as needed. A lot of the reasons why we even started kind of digging back into Perkinsea in the first place is because we had these screened in cages that you can see here in the bottom right that we were using to individually monitor mortality rates across individuals that were being released into these sites. And that's where we were finding that that's when they were getting infected. And we could monitor these infections and have a better understanding of what's going on. And just to put a little bit of a plug out there, we are trying to foster a bunch of collaboration across multiple stakeholders, both management, academic, whatever. Where a lot of these large-scale problems, they really need numerous people from a variety of different backgrounds to help address them. We are actively looking for people interested in collaborating with us to work in this Perkinsea working group. If you're interested, go ahead and contact me there. Now, finally, the most important thing, which is why I saved it for last, is that we need to dramatically increase our Perkinsea sampling. Ultimately, the reason why this is so important is because we need to really see where this thing is occurring to have a better understanding of how to diagnose and ultimately prevent outbreaks going forward. The surveying can take advantage of a lot of different rapidly developing molecular techniques to increase detection. And so there's a lot of promise here that even if you're not super familiar with some of these things, it makes it fairly easy and ultimately will make life a lot easier as far as being able to detect these things. Because unfortunately, outside of histology or genetic-based approaches, it is really hard to diagnose these infection outbreaks. With that, I will turn it back over to Vanessa.
Great, thanks. Okay. Okay, can you see my screen? Great. Okay, yeah, so I'm going to talk about now increasing Perkinsea sampling. So to better understand Perkinsea infection dynamics and its effect on amphibian populations, it's necessary to increase sampling efforts. Recent technology has made it easier to screen for pathogens, especially in the wake of COVID. So one of the systems that I'm familiar with is the BioMeme portable qPCR system. It's designed for the field. You can tell because it's been so strategically propped up in this field of grass here. So we use this system to develop a duplex qPCR assay to distinguish PPC and the putatively non-pathogenic NAG1 clades, and we validated it on the system. It has 3 wells, 2 channels, and an iPhone interface that docks into a handheld thermocycler. This machine was actually discontinued in 21, and they moved on to the Franklin, which has 9 wells and up to 3 channels. It's pretty user-friendly. It starts with a simple extraction kit. It's a 10-minute lysis step followed by a combined 3-minute transfer between color-coded reagents using a syringe that contains an extraction column. And then the eluate is transferred to tubes containing qPCR reaction components. It's run on the portable thermocycler, and the results can be analyzed in a cloud portal. I validated the system using tadpole liver tissue, which is obviously very invasive and somewhat cumbersome in the field. So it's much more streamlined to use swabs and eDNA filters. And that's exactly what Komarov et al. Used for presence-absence testing for BD in the field. They collected eDNA and skin swabs, stored them in ethanol, and later analyzed them in the lab using a Kaigen kit extraction and BioRad benchtop qPCR. From the same sites and individuals, they collected eDNA and skin swabs and then extracted and analyzed them in situ using the BioMeme end-to-end system. And they found that the swab technique had higher mean probability of detection in both the lab and the field. The lab-based system had a higher probability of detection for both eDNA and swabs. However, the BioMeme system was still able to detect all BD positive swabs in at least one of the 3 technical replicates and in eDNA samples at the same number of sites as the lab-based system. So it still offers a promising alternative to the lab. And this is good news since it's been confirmed in the lab that eDNA and swab sampling are effective means of detecting Perkinsea. So Matt kindly shared these figures from 2 manuscripts in preparation. In the first study, they compared the effectiveness of eDNA detection using 2 techniques, filter and sodium acetate pellet, and they compared it to tissue-based detection. Although Perkinsea was more frequently detected in tissue, the eDNA technique was effective with higher Perkinsea intensity in the environment. And then in the second study, they compared the effectiveness of swab versus tissue-based detection, and they found similar rates of detection between the 2 techniques, specifically in pre-metamorphs, and this is the stage that's typically associated with disease. So there's other portable qPCR machines on the market besides the BioMeme Franklin. Again, thanks to that post-COVID push for rapid in situ testing. I put together this table which compares some of the important specs of each system. So the BioMeme Franklin, as I mentioned, comes in a 1, 2, 3 channel model ranging from about 6 to 10K. It's lightweight, it's quite small, it can fit into the palm of your hand, as you can see, and has a typical runtime of about an hour. The Anatoa Maverick has 16 wells and a 2 or 4 channel model costing 6 or 12K. It's bigger and heavier than the Franklin, and it advertises a shorter runtime. Then the Chai Open qPCR also has 16 wells, a 1 or 2 channel model costing 6 and 8K. It's the heaviest and bulkiest of the devices. It kind of really teeters on the edge of portability, but it does have a very short runtime. And then there's the MIC qPCR, which has an impressive 48 wells with a 2 or 4 channel model costing 10 or 12K. It's relatively lightweight and quite small, and it uses this unique magnetic induction technology for rapid heating and cooling. So it has the shortest runtime of just 25 minutes. So it seems that the biomeme has some stiff competition. I will say that the biomeme does have the advantage of being the first to market. So it has had time to be independently validated by multiple research labs. But now qPCR systems, they have potential competition from CRISPR-Cas systems. A few years ago, I put together a proposal for a CRISPR-Cas-based system for detecting Perkinsa and other amphibian pathogens in the field. The setup looks like this. First, you collect either a swab or eDNA sample, then you add the swab or filter to lysis buffer. You transfer the lysate to a one-pot reaction tube containing RPA and CRISPR-Cas reagents. So this isothermal reaction is the major advantage over qPCR because all you need is a heating block, or you can just hold it in your hand, if you have enough patience. The reaction combines a recombinase polymerase amplification to amplify target DNA and then Cas enzyme cleavage of a collateral fluorescent probe. And then a detection strip with antibodies that are specific to the fluorescent reporter is dipped into the reaction tube to give a presence-absence result. An alternative detection method is using a UV lamp to detect fluorescence. And this has the advantage of providing coarse quantitation by comparing fluorescence intensity with known standards. And this was the detection method used by Honing et al. When they validated the CRISPR-Cas system for rapid detection of BD from swab samples. They generated a concentration-dependent fluorescent profile that had an LOD around 26 ITS copies. And they also compared a kit-based extraction protocol with a quick field deployable protocol. And unfortunately, they found that the kit protocol was sort of inconsistent in terms of technical replicates. I mean, at least one of the three, I think, got it every time. But again, you need 3 technical replicates, whereas the Kaigen-based, the lab-based protocol was much more consistent. And so this means that you're more likely to have false negatives when using that sort of field deployable protocol. The same group also validated the system for B-cell swab samples, but again, they ran into the same problem with the quick extraction protocol being less sensitive than the kit-based protocol, and in some cases, yielding false negatives. So ultimately, it really just comes down to focusing efforts on optimizing a field deployable extraction technique, because the CRISPR-Cas detection system, the qPCR detection system, those have been validated. So but you need to start out with sort of really high quality source material going into those protocols. But once you have that, it's really just a matter of customizing it based on the pathogen that you're interested in. So Perkinsa surveillance, it can be enhanced obviously by by these advances in field-based molecular sampling techniques, but it can also be enhanced by encouraging the participation of non-scientists in the form of citizen science projects. Surveillance data are typically collected by a small number of academic research labs and government agencies. Citizen science projects enable interested members of the public to voluntarily collect samples to support research and management. These projects have been successful in the past for BD. For example, the Sam Noble Natural History Museum in Oklahoma enlisted students in elementary through high school to swab amphibians throughout the state. And over the course of 4 years, they collected over 800 samples from 22 species across 23 counties. And they found about a 12% BD prevalence. So my PhD research helped support a citizen science project in the UK. After we reported that mortality event in those captive bred European tree frog tadpoles, the Royal Society provided funding for Tadpole Doctor. Tadpole Doctor was advertised on social media, and it was featured at the Royal Society Summer Science Exhibition in 2022 to encourage engagement from schools, zoos, frog breeders, and just general members of the public. Participants were provided with sampling kits and instructions for delivery, and the group received tadpole and water samples from 66 participants across the UK, and those are currently being processed at the University of Oxford. So I could see something similar being done for Perkinsea in the US. This could dramatically expand surveillance efforts, and it could provide a path for sampling private properties, which would otherwise be inaccessible. And I'm going to turn it now over. Oh, no, I think now we're at the, we ready for questions now?
Yes, we
Okay.
are. So we only had one question in the chat. So we'll go ahead and start with that one, and then I can unmute everyone or give them the ability to unmute themselves. So if anyone wants to raise their hand and ask a question, they can. But the question that we have is from Kirsten Nelson, and she was wondering if Matt or Vanessa, you could discuss the disinfection techniques a bit further. So specifically, she's interested in knowing if items or gear needs to be submerged in 10% bleach, and if so, for how long? And then how long would the item need to be frozen and at what temperature? And then a third question is, would you need to use both bleach and freezing to properly disinfect an item, or would just using a single method work?
I think that's one for Matt.
Fair enough. So one thing I will be happy to kind of comment on that is you do not have to use both of those methods at the same time, although probably couldn't hurt if you were kind of in that vein of things. So as far as the freezing goes, I'm not exactly sure on specific times of how long. We've just documented that after about a period of about 2 to 3 months, we don't see that those spores are viable. However, if they're kept at fridge temperature, that's enough to usually keep them viable, unfortunately. But to get more specifically into bleach, because I tend to think that that's a little bit more practical. What we've been doing historically is we create a 10% bleach solution using that really highly concentrated pool bleach, and we fill it into bleach sprayers, and we will spray that down on all of our field equipment and just let it sit for 5 minutes at a time. You don't have to completely dunk it, although that's probably going to be a little bit more thorough. However, one point that I do want to make is that before you do that, you really need to make sure you kind of scrub things off and get all of the detritus off of it. So a lot of like the pond muck and mud and all that kind of fun stuff. If you don't totally get rid of that, as kind of say Vanessa mentioned, you may be leaving some of those hypnospores that are hanging out in that soil clod kind of to be potentially problematic later on. I will say it is pretty harsh on field equipment. And for the most part, we go through dip nets and that kind of stuff pretty regularly. So it's one of those deals where, especially if you have a site that you know is Perkinsea positive, I would focus on maybe leaving some of your junkier field equipment for that site specifically. And so that way, those are the ones that are going to ultimately kind of get broken down anyways. And then focusing on using your other stuff and keeping that away from those Perkinsea positive sites.
Awesome. Thank you. Kirsten, if that doesn't answer your question fully, please feel free to speak up. I'm going to allow you guys to unmute yourselves now. We have another question in the chat. I believe this one is for Vanessa. This is from Pedro, and he said, what is the dropout rate for the voluntary surveillance?
Oh, yeah, that's a good question. So you mean like how many people who sign up don't actually like submit samples? I don't know. I'd have to reach out to I would have to reach out to the group at the University of Oxford to get that exact number. I think that, I mean, people generally seem pretty excited about it, especially like when there's kids involved. So I would imagine it's probably not very high, the dropout rate, but again, it's hard to say. But I think that if you have the right pitch and you're reaching the right people, you can get people pretty excited. I mean, everyone loves frogs. They want to help out. They want to help the environment. So it's not hard to convince them to be a part of it.
Awesome. Thank you, Vanessa. Does anybody else have any questions? I just want to remind you too and direct you to the chat that Carrie has posted our Wakelet, where you can find a whole boat of resources about Perkinsia that Vanessa and Matt have put together, as well as myself and Carrie. Another question in the chat from Amanda Starr: is there a ddPCR method for Perkinsia?
Oh, ddPCR. I haven't heard of one, but there's no reason that it couldn't be adopted for it. Have you heard of anything like that, Matt? ddPCR? It's like high throughput PCR.
Potentially, maybe some of the stuff that was done like pre my time, but honestly, I don't know.
I mean, that would be something I think that would be advantageous for a project that needed a high degree of where you needed to sample a lot of individuals. Because I know that the field-based methods, because I mean, at most you have maybe 48 wells in one of those qPCR machines. So they're great for getting to remote places and getting your answer quickly. But if you're looking for a lot of data from a lot of different individuals, then using like a benchtop and potentially ddPCR would have that advantage.
There is a more general question in the chat. Amay is asking if there's any tips, or either of you have any tips on how to find local openings for work or shadowing on work with amphibian diseases to better prepare for continuing their degree. So I'm not sure if you have any resources to share with them.
So there's definitely a lot of different opportunities. I know SSAR, the Society for Study of Amphibians and Reptiles, is doing some more kind of here's how to find grad school approaches. They're actually doing a webinar on that a little bit later this evening. What I'd actually really recommend is getting involved with your local park chapter. I've been a part of PARC now for 10 years and heavily involved with their disease task team. They put on workshops on how to do dissections and how to do some of the histological examinations, things like that. There's often regularly these panels on how to progress in your career as a herpetologist or a disease person. So honestly, it's also a great opportunity just for general networking. I very, very strongly push a lot of my undergraduates that I mentor to go to opportunities like that to be able to kind of get involved, get a little bit of the technique aspect of it, but more so find the kind of networking opportunities that ultimately lead down the road to kind of their ability to do a master's, a PhD, or just be more involved in that field.
I'm actually looking for the link for the SSAR information session now. All right, I actually found it.
They also, just as a heads up, do a really awesome Google Sheets that keeps track of what labs are actively soliciting grad students. It's just a good, quick and easy way to at least start looking and then use that to kind of go that direction.
Awesome. Yeah, so it looks like you can just go to that website that I just posted in the chat and you can register. And it is tonight at 6 p.m. EST. All right, are there any other questions? All right, well, if there's no other questions, I'd just like to take a moment to thank our speakers. Again, this has been recorded, so it'll be up on the PARC Disease Task Team YouTube channel, which you can access through the PARC Disease Task Team website. I'm going to go ahead and stop recording now, but if anyone has any last minute questions, I'll just kind of hold on for a sec. Thank you again, Vanessa and Matt. That was an incredible presentation.
Thanks for having us.
Also, just want to do a quick shout out to the amazing work that Jenna and Carrie Wickstead and other members of the task team put together to organize all of this and help us, people that aren't quite always the most technologically savvy, get through today's meeting. So thank you all for your energy on that.